going to talk really quickly slides, but before I do, I would like you to do me a favor and go over to your workspace that you've been working in, and at the top right, you're going to have a notebook runtime, and right now yours should have a triangle for play arrow, and I want you to click it so that your runtime starts up, and because it takes a few minutes, so while I'm talking through the slides, it will be running, it will be spinning up, and hopefully by the time we get to the hands-on part, you will all be able to work on it. So if you have any issues or it's not working for you, please write in the chat and someone online will help you with that. Okay, so I'm going to talk to you about the second kind of analysis, which is yes.
Sorry, can you show your screen one more time so that people can see where you click the runtime?
Oh, yep. Do you see up at the top right?
Yes.
Everyone see this at the top right? Where it says no, mine is running. So yours will look a little different. And because I started mine up in advance, and you'll just want to make yours go. Okay, it's like,
do they need a particular runtime environment?
Well, that's a good question. Do you have a particular one? Well, no, the default will work fine. They're going to have to recreate it for yours, right?
Yes, but we'll go.
Okay.
I just wanted to make sure.
Yep. So the default is fine for that, for our purposes. Okay. All right. So I'm going to go back to. Move this. Okay. I'm going to talk about interactive analysis. So what is an interactive analysis? We're going to have a very brief overview of this second kind of analysis in Terra, and then we're going to do a hands-on, so you'll get to actually see the analysis in action. So we've done this first part, running general automated bulk workflows to do data processing, and we're moving on to the second part of the analysis. This is more specific and manual. Maybe you want to interact with your process data or look at it, visualize it. So when you're done with this stage, it usually addresses the hypothesis, and you'll get some scientific conclusions, which you'll then be sharing. So we have two forms of interactive analysis on Terra right now. We have a Jupyter Notebook and we have RStudio. I'm going to talk about Jupyter Notebooks, and Liz is going to follow with a different use case, and I think talk about RStudio as well, which is a new feature in Terra. So this is what a Jupyter Notebook looks like. Can I get a heads up for, has anyone not used a Jupyter, not heard of or not used a Jupyter Notebook before? I actually can't.
Haven't used it.
I'm sorry?
Yes, Allie, I haven't used it.
Okay, so notebooks are, this is what it looks like, and you'll see that in a second. The nice thing about notebooks is it includes code and documentation all wrapped up in one package. So the nice thing about it is it makes you, if you write enough documentation, it makes your work very reproducible and shareable. And it's easy to share your notebook with another colleague who can look at it. And I think it'll be clearer as we do the demo, but I want to get to that because that's the very satisfying part. So in Terra, your notebook runs on a virtual machine or cluster of machines that we call a runtime. And you have a couple of choices. So you just started your runtime and used the default example, but there's a lot of options for customization. So when you click on the gear icon, instead of on the triangle play, you get a configuration form which lets you choose the environment. We have a number of different built-in environments. These are just the libraries and packages that are loaded by default. And it also will allow you, I'll show you this in the in the walkthrough will also allow you to have a custom environment, and you can use a custom Docker image, and that's super useful for reproducible work. In addition to the environment, you choose what your power is, how much CPUs, memories, RAMs your virtual machine or cluster of machines has, and there you have the option of different profiles, and you can specify all these down here. And I'm going to not spend a lot of time, but go through it in the walkthrough. So this is a little bit more on how to make your own image. We have documentation for that in our how-to and tutorials guides. Very quickly, I want to introduce a tool called Hail, developed at the Broad. It's a Python-based library tool that allows you to work with large genomic data really efficiently. And it's an open source framework that analyzes the largest genetic data sets and to meet the needs of hospitals, diagnostics labs, and industries. This slide gives some resources for the Hail website, and this is who it was developed by. It's Cottonseed and John Bloom from the Broad. And this is what it gives you. It's for people who do GWAS. It's a very efficient and interactive, so efficient meaning saves you time and saves you money, way to do analyses on tens or hundreds of thousands of samples. And I'm not going to spend a lot of time because we have a great workspace that's very self-guided. This link will be available in your slide deck, and I encourage you, after you get a general introduction to working with notebooks, to go and check it out. It has a couple of has a GWAS tutorial that you can walk through, and it has a mock data tutorial. So I just wanted to introduce people to that as a little bit of a teaser. Some of you may have heard of Hail, and this is a great resource that we have available. So I'm going to switch super quick over to my workspace. I actually created this one yesterday. You're probably on your job history page. Mine is succeeded because I started it yesterday. And I would like you to hop over to the Notebooks tab. Is anyone's Notebooks tab not running? Notebooks runtime, pardon me. Can you say that in your chat? If it's not, I would encourage you to follow along with mine and to go afterwards through your own notebook. Notebooks are pretty self-explanatory because they have a lot of documentation, and that would be better than trying to get yours running at the same time. So there are two notebooks in this workspace. We're going to open the Explore Phenotypic Data. The Teradata Util notebook is just a list of libraries, or sorry, it defines functions. So when you click on a notebook, you get a read-only preview, and you have two choices: to edit the notebook, or you can be in playground mode. Playground mode allows you to run the notebook, but you can't save the data. Or, sorry, pardon me. I misspoke. You can't save the notebook if you make any changes to it. The reason we have playground mode is if you have multiple people working in the same workspace at the same time, you don't want one person to do some analysis in a notebook and save it, and then another person comes along at the same time doing an analysis and saves it and overwrites the first person. So only one person can be editing a notebook in real time in a workspace. So you're all on your own workspaces. You can click edit mode, and it should pop up pretty quickly. If your table of contents doesn't show up on the left, you'll want to click this little table of contents icon under the help menu. I'm going to really briefly give a few pointers on how to work with notebooks. So notebooks have two kinds of cells. They have documentation cells, which are these ones with text. And if you scroll down, there are code cells. So to run a cell, let me back up. To edit a documentation cell, you double click in it, and then you are given the code in Markdown, and you can edit it if you want. And to finalize that, you click the Run button right here, or you can also, a key shortcut is Shift Enter. So anytime you're in a cell, you can click Shift Enter to run the code. So this has a lot of information about the data that's in this workspace, which I encourage you to read about. And the first step of any notebook is to set up the notebook. Often there's some additional libraries or things that you'll want to use above and beyond what's in the notebook environment, and this is where you would add those. Runtime values will tell you what you want to be running if the notebook analysis needs a particular size notebook or size runtime. Maybe you need a Spark cluster because it's a particularly large analysis. That would be in here. And you always want to change the notebook runtime first before you do any work, because when you change the runtime, many of the variables and data that's stored on the local virtual machine is lost. That's going to change soon, but that's the case. So basically, you want to run the cells that are code cells in order. So this is the first code cell, and to run it, you can click the run button, or you can click Shift Enter. When it's running, you'll see there's a little asterisk beside this in these brackets, and then when it's completed, it will turn into a number. So, and the number represents the number of commands that have been run in this runtime since the beginning. And you may have noticed that the table of contents turns red as the code is running. So I'd like you to actually go through and just run the code really quickly, because I'd like to get to the part where we are actually doing a plot. So here's the section where we're going to load the phenotypic data from the data table. So the data's in your workspace. It's in the workspace data table, the phenotype data, but it's not in the notebook computational environment. So this section is where you're going to do that. The first thing you need to do is you're going to set the environment variables. So like the Google billing project that's associated with this workspace, how are you going to pay for it? What's the name of the workspace? And this code cell has been collapsed up because it's not really important that you know how to code these some of these parts. If you want to see the full code, you can click to expand. So you can run that, and it will print out the billing project, which agrees with what I see at the top, the workspace, which is what I have at the top, and this is the Google path of the storage bucket. So in this section, we're going to load the phenotype data. First, we're running the utilities notebook to grab some of the commands that are in that, and it's going to access from the table, the subject table in your workspace. This one, it's going to pull the phenotypic data.
Sorry.
Yes. No, it's fine. Yep. Was someone? Am I missing?
I've wrote that I'm getting an error when I'm trying to run the third.
Yes, the third one, like creates an error of a file not found.
Oh, you know what? I just realized. Okay, so file not found. What's that? What's that? I am thinking if you look in the notebooks in the workspace, I'm going to make open a separate tab. So I'm going to look in the notebooks. No, that's what it's named. It's not, is it not finding this, does it say which it's not finding?
It's that one, Terra data util dot
notebook.
IPY, yes.
Is it, can you check in your notebooks tab and make sure it is there?
I see the Terra data util. Do I need to turn it on or something?
No, it's supposed to run it. I'm wondering if someone on the chat can help, because
Yeah, I'm thinking, Allie, what this could be about. Some people are able to run it okay, and some people are running into issues. So
that is odd.
Right. So yeah, the cells for this need to be run in order.
Oh, yes, yes.
Okay.
That's true.
So,
because it needs to find that, because this is where it's finding the workspace bucket in the one above this. What happens if you click on the one that's not running, go to cell and try run all above and see if it runs.
I get a different error of consolidated table.
It's not finding the consolidated table name. No, that's actually, no, that's defining the consolidated table. It should.
okay. Let's see here.
So it's supposed to be defined in the Terra data util one, right?
What is supposed to be defined there?
The consolidated table and
no, this is just a variable that so this is a variable that's used later, and it's so if you notice in the next command where it's getting the Terra table, and see how it says consolidated table name. So this is
there you go.
Yeah. So right here underneath the run Terra data util, it's just defining it's just defining the table that you're going to read from, and it's the subject table which has table.
Okay, I've managed.
Okay. Okay, should I keep going?
Yeah, I think for the interest of time, let's keep going.
Okay.
We can figure
Yeah.
out. Yeah,
okay. Okay. Yeah. Okay. So this gives, this just tells you that you've imported, if you were to compare with the data table, if you looked at it in the data tab, this should be the same information. This just changes the first column and it will print it out again. And then this is the section that gives you a little bit of an idea of what you can do with an interactive analysis. So there's a lot of documentation, but But basically, we're going to define 3 plotting functions right here. We're going to define a distribution plot, a bivariate distribution plot, and box plots. I'm not sure why it's giving me that error. And in the next few, it basically allows you to go in, it's preset with some information that will give you plots of the phenotypic data. So this is just not scientifically terribly interesting. This is just to give you an idea of what the plots look like. So here I'm plotting the low density lipids from my 2,504 participants, and it looks like we would expect, right? And I have defined another variable, the BMI, and it looks like you would expect. And then same with bivariate distributions. We have the BMI baseline and the height baseline. And I don't actually want to spend a lot of time, because what I'd like for you to do, and I know we have a break now, and you can do this into your break, or you can take a break if you need to. But the idea is, you can play with these variables. You don't really have to know how to code in order to use a lot of the notebook functionality. You know that, these are the variables, and if you needed to change, if you wanted to change the variable, you could look back up where we plotted out the entire table, and you could say, oh, well, I wanted to plot by HDL, or I wanted to use the population in my box plot. So this allows you to do that. I know I've run over, and I know we have a break, and I want to give people a chance to have a break. So I'm actually going to stop here, but I'll stay online and Liz will be up next. Does that sound okay to everyone? I know this was a very quick overview. At the end of this slide deck, there's a reference. We have a Notebooks Quick Start workspace, and if you haven't used Jupyter Notebooks before, it's a great place to get started. It's a very well documented workspace that goes through everything from how to type in a notebook to how to do analysis. So I'm going to stop here, and if anyone has a question, we'll go till at 10. We'll do 10 minutes, and we'll cut the wrap up short, I think, but leave Liz her full 45 minutes to talk about bioconductor. Okay. So I'm going to.
Thank you.
Yeah.
So it looks like we have a few questions in the chat, Allie.
Okay. What would it be better for me to have on the screen? My notebook, so I can maybe help? Let me see.
I think that's a good place to start.
Okay. Why am I not seeing my chat? Hold on. Oh. Okay, yeah, we have quite a few questions. Okay. Okay, so I'm going to address some of these out loud if I can. So we have a question about box plots error. I get this error. Key error, demographic annotated sex. I tried to solve it, but I could not. So usually when you have something that references what you think is a variable, the problem is the variable name is mistyped, or in this case, I happen to know that what happened was I took this notebook from a workspace that the variable was called demographic annotated sex, but in this workspace, the variable is just called sex. So if you go up to that, if you go up to the code piece here, and where it says, so you're getting this error, but if you replace it with the right variable name, it will give you the right thing. So as with computers are not smart, and notebooks are no different, and they can never make the jump between what you typed in and what the actual variable name is. So oftentimes, I always make it a practice to check the variable name first. Reference link. I will type in the so if you want to look for the notebooks quick start, you can look in the Terra notebooks, look for the Terra notebooks quick start in, it's in the showcases library. I'm looking back. Yeah. Here, you know what? I will get the link for the notebooks quick start to send. But are people able to get the graph going? I'm hopeful. So I'm going to go to examples, and it's a featured workspace. Here's the Hail Notebooks tutorial right here. I'll get that link out for you. And continuing down. Here's the notebooks quick start. It has a lot of documentation. There's several notebooks, including an intro to Jupyter notebooks, which is it's a great first primer if you've never used Jupyter notebooks. Has shortcuts and things that can trip you up. And I will send that link around as well. And you can run everything in that workspace, and it costs, I want to say $25.50, less than $1. These resources are also in the slides. Are there any other pressing questions?
No.
Am I good?
Thank you.
Yeah, sure. I'm sorry it was quick, but the notebooks quick start and the Hail workspace are they're great for self-guided. They have a lot of documentation. It's pretty clear. So hopefully you'll be able to. I've given you a taste, and Liz will give you even a better taste, and hopefully you'll be able to go from there. So I'm gonna.
I do actually have one last question. Does it
Okay.
does the notebook turn off automatically, or are you supposed to turn it off?
That is a good question. I didn't talk about cost. I apologize. So the notebook will turn off automatically if the browser is if there's no activity in the browser for I think 20 minutes, it might be half an hour, and if there's no calculations being done. So if the browser page is if you click in the browser, it will not turn off. or if there's an analysis calculation running in the background, it will stay running. So those are all desired behaviors. You can get information right up here at the top about how much it's costing you. Notebooks cost no matter what they're doing at the time. So if I were to go, so I'm not actually running any of these notebooks right now, but my runtime is active and it's costing me money. That's why we have an auto pause feature that pauses things when you're not active or when there's no calculations going on. Is it possible to run 2 notebooks simultaneously? I do not think so, not in the same runtime. I don't think so. But I could be not right about that. You could post that to the support. They would be able to better answer that.
Allie, just thinking about the notebook you just demoed, it did run the Terra Utils notebook at the same time as your other notebook was open.
Oh, that's true.
So perhaps if it's on the exact same runtime, if the notebooks have the same requirements, it's okay. If you had to run a Python notebook and then an R notebook on that had 2 different environments, I'm not sure you could do that. I think you would get a warning about your runtime not being the appropriate one.
You're right. You're right, Liz. Yeah. Do you want me to stop sharing and pass it over to you?
Yeah.